Em nghe không?
Oh, hi everyone. Hello everyone, and welcome to the SEC Club this week. So for this week, we're excited to kick off this session on exploring the Tahoe 100 million dataset. This atlas represents a huge achievement in single-cell biology, encompassing 100 million individual cell transcriptomes. And my name is Hazel. I'm a scientist at Titering, and joining me today is Julie, also a scientist here in Titering. Today, we're diving into Tahoe 100 million, which provides the responses of 50 cancer cell lines to a staggering of 1,200 drug perturbations. As you can see on the right here, the Disney plot gives you a glimpse of the scale and the complexity of this data, highlighting the diverse cell populations and the over 60,000 unique drug cell interactions captured. And our main goal today is to demonstrate how you can leverage talk to data to explore, to analyze, and to gain values, insights from this remarkable data set. Okay, and this is the agenda today.
Yeah, so I'll give a quick overview of what we'll be covering today. So first, we're going to start with a brief introduction to the Tahoe 100 million dataset. We'll discuss its key features, the experimental design behind it, and why it's such a valuable resource for single cell research. And this will provide some important context before we jump into the practical part. So then the main focus of our session will be on exploring that dataset directly within the Talk to Data platform. And so we'll demonstrate how you can navigate the data, perform analyses, and visualize the results. We'll cover some specific use cases and show you how to leverage Talk to Data's tools to extract meaningful insights from this massive dataset.
Yep. Okay. Now let me go ahead and introduce you Tahoe 100 millions. Okay, so as you can see from the author list right here, this was a collaborative effort from researchers at Vivo Therapeutics, the ARC Institute, and Past Bioscience. So at its heart, Tahoe 100 is a single-cell atlas. So it's one of the largest publicly available single-cell dataset to date, encompassing like 100 million individual cells, transcriptomics profiles, and but it's just not about the size. The dataset also boasts incredible breadth and depth, covering 50 different cell lines that were subjected to over 1,000 small molecule perturbations. And this allows us to study how these perturbations affect the gene function in a multitude of cellular contexts. And essentially, this dataset is a very powerful tool for unraveling some fundamental principles of gene regulation and network dynamics. And also because of its scale and diversity, is ideally suited for training robust AI model in system biologies. And this has some potential to advance our ability to predict and to manipulate cellular behaviors. And also, beyond the basic research, this dataset also offers very, very valuable insights for cancer drug discovery. Okay, and now we'll go ahead and explain you how the data was generated in this data set.
Yeah, so before we jump into any analysis, I just want to go over how the authors were able to generate this large of a data set here. So the researchers developed a platform called Mosaic, as you can see illustrated here, and the process begins with these mosaic tumors. And so these are spheroids or 3D cell cultures composed of a mixture of 50 different cancer cell lines, and they created what is called a cell village. Now, this approach is crucial for minimizing batch effects, ensuring that the variations in the data are due to the experimental conditions and not just random differences between samples. Because for those of you who have grown cell lines before, typically you'll put one individual cell line into a single well in this 96 well plate, but instead what the authors did is they combined all 50 cell lines into one well here. And so they seeded these mosaic tumors into the 96 well plates, which is just standard format for the high throughput experiments. And then each of these wells also received a specific drug perturbation, meaning that each spheroid is treated with a different drug or a different dose of the same drug. And more importantly, DMSO vehicle controls are also included as well. And DMSO is a common solvent used in drug delivery. And so these controls help to isolate the effects of the drugs themselves from any potential effects of the solvent. After treating the spheroids with the drug treatment, they waited 24 hours, and these spheroids are then dissociated or broken down into the individual cells. These single cells are then barcoded using the Parse GigaLab kit. And this is a crucial step because the combinatorial barcoding allows researchers to uniquely label each cell so that they can trace it back to its specific well and the cell line for later analysis. Okay, and then finally, the barcodes, along with their known genetic information or the genotypes of each cell line, are used to deconvolute the data, which means that the researchers can determine both the drug treatment that the cell received and the cell line in which it came from. So, in essence, this mosaic platform with its cell village approach and genetic demultiplexing is the key to generating this type of massive data set. And so it minimizes the batch effects and allows for simultaneous profiling of thousands of conditions at a single cell resolution.
Yes, that's very impressive. And then now we go ahead and view a little bit the actual genetic landscape of the cell lines used in this study. So as you can see here, this is an oncoplot. This provides like a representation of the most frequently observed genetic mutations. As you can see on the left here, those are genetic mutations across the 47 cell lines that were well presented in the dataset. So as Julie just mentioned, this dataset has 50 cell lines. However, there are 3 cell lines which were, is just too low in cell numbers. So we have 47 left that were like really well represented. And as you can see in this plot, these cell lines are derived from 13 different organs, with the majority originating from lung, bowel, pancreas, and skin tissues. And this diversity in tissue origin is important because it allows us to study drug responses in a variety of cancer. Why? Because you can imagine that each of the each of the tissues here representing one type of cancer. For example, we have lung cancer, pancreas cancer, and skin cancer. Okay, and the plot also highlights the present a wide range of driver mutations on the left here. So driver mutations are genetic alterations that play a crucial role in cancer development and progression. Notably, we can see some genes like the TP53, the CRAS, the CDKN2A are altered in about half of the cell lines. So each square on this chart is one cell line, and you can see that these 3 genes mutation appear in many of the cell lines. Okay, so this also indicating their importance in this cancer cell model. Okay. And this genetic diversity is a key feature of Tahoe-100 because later on we will discover how these mutations affect the drug response, and it allows researchers to investigate how different genetic backgrounds influence the drug responses. And by combining this genetic information with the drug response data, we can explore how specific mutations impact the efficacy of different treatment. And to sum up, this oncoplot provides like an overview of the genetic landscape of the cell lines used in the study, and it also set the stage for subsequent analysis of drug-induced transcriptomics changes and allow us to explore context-dependent responses to various perturbations. Yeah, in here. Okay. And Okay, that's an overview about what mutations can we see in the cell life. And now we can shift our focus to the drug perturbation themselves. We'll see what kind of drug we have and what are the targets.
Mhm. Yeah, so in this data set, the authors included almost 400 small molecule compounds used to treat each of these cancer cell lines. So in figure 2b above, this provides an overview of the mechanisms of actions or MOAs of these drugs. So in total, there were 379 distinct drugs used, and 180 of them could be classified into 25 different MOA categories. As you can see, some of these MOA categories are well represented, which indicates which pathways are most frequently targeted. So, for example, we see a significant number of drugs. Let me turn on my mouse here. Drugs targeting pathways such as the PI3K, AKT inhibitor, as well as the HDAC up here, and proteasome inhibitor as well. It's also worth
Mhm.
noting that the majority of these drugs, about 69%, are approved agents, which highlights the clinical relevance of this data set as well.
Mm.
Okay. And so moving down below to figure 2C, we can see the specific genes which are targeted by these drugs. And in total, these drugs reportedly targeted 325 genes, with the top 120 shown in the figure here. I think what's particularly interesting is that 120 of these drugs are targeted by, I'm sorry, 120 of these genes are targeted by more than one drug, which suggests the potential redundancy or opportunities for combinatorial therapies. And this is crucial information for researchers looking to develop more effective cancer treatments. So basically, in essence, these figures provide valuable insights into the drug perturbations used in this data set. They highlight not only the diversity of the MOAs and the target genes, but also the clinical relevance and potential for combination therapies. And so basically, this information is crucial for understanding the scope of this data set and for designing experiments to explore drug responses in different cellular contexts.
Okay. Okay. Now that we kind of like understand the background, this data set, what cell lines are included, what kind of mutation, what kind of drugs, and what kind of target. Now, now we can move on and start diving into a specific result presented in the paper. So we're beginning with figure 6A in the paper, if you want to look it back. So this figure right here examines the effect of dabrafenib, a BRAF V600E inhibitor, as you can see here. So on the expression of a RAS-RAF pathway gene signature. And as you can see on the left right here is the actual the actual pathway. And oh, I'm sorry. Okay. So to understand how this drug works, we need to consider the RAS-RAF pathway first. And which like in here, the pathway is critical. This is like the MAPK, the famous one. And this is a very famous signaling cascade that controls the cell growth, the proliferation, and the survival of the cells. And is frequently dysregulated in cancer, making it a common target for therapies. And as you can see, the pathway start with growth factor activating receptor like the EGFR2 here. Once it receives the signal, it will lead to a series of protein interactions involving the RAS, the RAF, the MEK, and the ERK protein, and ultimately affecting the gene expression. Okay, and in this experiment, the cell lines, they were categorized into three groups depending on the mutation of either RAS or RAF protein. Okay, as you can see on the left side of the figure 6A here is driver mutation status. So it can be wild type, which means no mutation in both of the protein. It can be mutation in RAS protein and it can be mutation in RAF protein. And both of the mutations, they are GOF or gain of function mutation, which means that it will make the protein constantly active. And in here, you can see if it's the RAS, like the RAS gain of function, right? So which means these cells has a gain of function mutation, in the cross genes. The cross gene is the gene encoded for RAS protein, and is similar for the RAF gain of function mutation. Okay, and then the violin here. The violin is the main point of this finding. The violins show the effect of dabrafenib at different doses, from lowest dose to medium to highest dose, right? And as expected, dabrafenib, which specifically target the RAF V600E mutation, as you can see, RAF, is specifically target the RAF, the green one, right? And as in the chart, it shows the most significant inhibitory effect in the cell line with RAF mutation, with RAF gain of function mutation, right here. And in contrast, it has a limited effect in RAS, in crust, and mutated cell lines where the RAS is the primary driver. Why we say is show the most significant inhibitory effect, because we can see that the pathway expression is down, is very lower, is significantly lower in RAF cells. Okay, and this confirms the expected behavior of the RAS-RAF pathway, and it also demonstrates how this data set can be used to study the context-dependent drug responses, and it also highlight the importance of considering the genetic backgrounds or the driver mutation of cancer cells when developing and applying the targeted therapies. Okay, and from here, this is one findings that we saw in the paper. From here, we will try to show you how we can replicate this finding on top of the data. But before we move on, I want to us to keep in mind that this is the finding. This is the main finding, that cell lines with RAF V600E mutation should exhibit the lowest MAPK expression, or like the RAS-RAF pathway expression, when treated with dabrafenib. That's like the findings and also the expectation from the paper. And now we go to Tahoe 100 million on top of the data. We try to reproduce that finding. Okay. So let me try to go here. Let me try to go here. Oh, I'm really sorry. I just accidentally stopped sharing my screen, but let me try to share it again. Okay. Try to share it again. Okay. So yeah, it's back. So Julie, can you see my screen now?
Yeah, we can see it now. You can see talk to data.
Yeah, okay. So this is actually how Tahoe 100 will look like on talk to data. As some of you here, you may know about the beta version. And with the release of Tahoe 100 million, we also release an update UI for the beta version. And you still will see. So these are 50 cell lines within the Tahoe 100 dataset. Okay, and within the dataset, what you can see are all here. So first, you can see different cell line. You can see different cell cycle phases of each cell line. You can see the cell source, or the cell source are the original tissues. You can see different kind of tissues like colon or lung. And then you can see different kind of cancer of these cell line. And then next, we'll see two things related to drug, the drug concentration and the drug name. Last but not least, we have the origin tumor site. It can be either primary tumor site, or it can be the site of metastasis, mainly lymph node in this case. Okay. And you can query the gene across different cell lines and different kind of drugs. You can combine different criteria metadata to explore the data set. But let me use an actual use case for you. Okay. So this is Tahoe 100 million, and this is how we can reproduce the effect of dabrafenib on the RAS-RAF pathway on top of data. Okay.
Yeah, so basically, the figure that we're looking at here, we were trying to recreate that last figure, which we saw in the last slide, where you'll see three different cell lines, which we have filtered out in the data set. The first cell line, SW48, is the KRAS mutated cell line. So this has the RAS gain of function mutation. The second cell line, HT29, is the BRAF V600E mutation, and so this has the RAF gain of function mutation. Lastly, the A172 cell line is the RAF-RAS wild type, so this has no mutations. And within these cell lines, we have also filtered for the drug name. So each of these cell lines have also already been treated with the dabrafenib drug as well. And when we query for a specific gene, MAPK1, we're able to see the expression level across these three different cell lines that are already been treated by the drug. And notably, we see the least MAPK1 expression in our HT29 cell line, which contains the BRAF V600E mutation.
Yeah, thank you, Julie, for some of the finding. Now I will try to show you actual working step on talk to data to be able to reproduce this chart. Now let me go back to talk to data. We need to visualize the gene expression on different cell line. So we will view by cell line first. We'll view by cell line. So what we need to do, we have this little filter button right here. Okay. Oh, okay. I'm sorry. And then we can select our three cell line, the HT29, the SW48, and finally the A172. For three of the cell line right there, we need to be treated with the dabrafenib, and that. Okay. Now we are looking at all 50 cell lines. We don't want that. We want to focus on the three selected cell line only. So I will apply the search first. When I apply the search, you can see that now the selected cell lines are highlighted in blue, but I just want to focus on them. So I will only select to view these three nodes. Now, the size, the node you see here is actually the number of cells for each cell line that are treated with dabrafenib. So the tree is already filtered by dabrafenib right here. And with A172, we have around 7,000 cells treated with dabrafenib. Okay. Now next, how we can include the genes here. Go to this search button and we just include the MAPK1 and search for the MAPK1. Okay. You can see the size of the node is changing. The size and also the color change. So when we include the gene into our search, it turned from gray to red, different kind of red. So the redness of each node actually represent the expression level of the query gene. The darker the node, the higher the expression level, which means in here, HT29 has lowest expression level of MAPK1. And the size of the node is the proportion or like the fraction of cells expressing MAPK1. And now in A172, not only is really highly expressed the gene, but also a huge proportion of these cells expressing the gene, comparing to the HT29. Okay. Now we go back to the slide, and that's how we reproduce this one. Okay. Now we move on. This is just gene query. We're investigating the expression of one gene, but how about many genes? We want like, let's say different MAPK genes. How can we do that? So here you can utilize our composition chart. Okay, I will first show you how to include different metadata information into the chart. Just in here, you can see beside the drug and the cell line, we also have different drug concentration. Okay, And I will also show you how you can include different genes into the chart also. But now we go back. Now we go back and we go back to the filter. Why? Because we need to include the drug concentration to our composition chart. We go here. We just like, for example, I just put in the low and medium concentration and I apply the search again. And now it's here. Now I just go to the composition and create a new analysis. It should start with the drug and then drug concentration and then the cell life. Okay. 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 And that's how we can reproduce the composition chart. Now let me go back to our chart. I'm sorry, I just forgot. So this is one gene, map K1. You can just type more gene here to add more and more genes to our composition chart. Okay. Okay.
Okay. So before we move on, we do have a question from the audience from Preston, who asks, how do you open this data set when you first get into Talk to Data?
Oh, yeah, I think we can wait 1 second and go back to the platform. We go here. Okay, right there. Oh, let me just go back to the main interface. Oh, so Preston, just to head up, this is a testing phase for this week, and next week, this data set will be available on Talk to Data beta version. And how can you open this data set, right? When you get into beta version, like Talk to Data beta version, you can hover your mouse to this setting button. Okay, in this setting button, you can select different database version. So for all of you here, next week, like the first week of April, we will release to you guys both the Tahoe 100 million dataset and our upcoming version 5 database. At the time, you can select Tahoe 100 as one database version, and you may notice down here, this is normalization function. This is also one new feature that we are testing on, where you will be able to select the unit, the expression unit of your choice. It can be either the Bioturing rank, the log normalized, or the raw count. However, this is just in the testing phase, and we are trying to figure out like what is the best approach that so that we can bring this kind of unit to you guys. And so yes, this is how you can access Tau 100 million on its release. And Preston, we will also email you as soon as the dataset is actually ready on your end, so you can start taking it. Okay. Okay. Now let me go back to the slide so Julie can continue with this composition chart.
Yeah. All right. Thank you. So again, what we're looking at here is our 3 different cell lines at 2 different concentrations treated with the drug dabrafenib. And not only can we look at the expression of MAPK1 across these 3 different cell lines, we can also look at the expression of other MAP genes such as MAP2K1, MAPK8, and MAP3K1. And overall, when compared to our wild type cell line, which is A172, we see a consistent decrease in the MAPK expression in our mutated cancer cell lines, especially in our BRAF V600E mutated cell line, which is the HT29 cell line. So just overall, we see a decrease or downregulation of the expression of our MAPK genes in the mutated cell lines.
In here, also there's one interesting thing we saw, that actually the doses of this drug doesn't really like affect the expression that much. And that's also one thing we can see from the original figure in the paper, that even with like different doses, high, low, or medium, the effect is, it changed not that much. So that's just one thing we noticed for now. Okay. Okay. That's like That's about the composition chart. Now we will move on to the next part where we want to actually, let's say, take a more quantitative approach to analyzing the effect of dabrafenib. Why? Because in here you can see the changes in gene expression. Yes, we can see, but we don't have any statistic involved. We don't know whether the changes are like really statistically significant or not. So that's why we perform differentially gene expression analysis or the DGE. So this feature allows us to statistically identify genes that are significantly up or downregulated in HC29 cells, which have the RAF V600E mutation when compared to the A172 cells, which are RAF RAS wild type. Okay, and now I will go to the platform so I can show you how did I set up this DGE analysis. So in order to set up the DGE, you just need to go to the differential expression button on the right here, and then we can select the two different cell lines for two groups. For the group 1, we should select the HC29, and for the group 2, we can select the A172. Both are treated with dabrafenib down here. Dabrafenib, okay, this one, and dabrafenib, this one. You can see that the Venn diagram, they are changing real time, reflecting the cell numbers in each of the group. In group 1, in the mutation group, we have the around like 6,500 cells. And in group 2 of the wild type group, we have around 7,000 cells. We can name the gene here, dabrafenib, down here, like that, and then run the analysis. Okay. And I already run one, I've run one experiment earlier. I can quickly show you how the result look like inside. First, you will have the differential gene expression table. And when you go to the full report, you have a volcano plot to visualize like which genes are up or down regulated in the HT29 cells. And let me go bring you back to the slide so we can see that when I search for the MAPK genes here, most of the MAPK genes are downregulated in here. You can see in blue, or let's say it has negative log2 fold chain values. They're downregulated in BRAF V600E or in HT29 cells. Okay, and also these downregulation are all significantly meaningful because they have FDR or the adjusted p-value lower than 0.05. And up here above the violin plot, we have above the volcano plot, we have the violin plot to represent the expression level, the distribution of these selected marker genes between like the two groups. Okay, and this DG analysis provides like a statistical evidence that dabrafenib significantly suppressed the expression of marker genes in HT29 cells, further validating our previous observations and the findings from figure 6A. And in here, by performing DGE, we can identify the key genes. And eventually, we can also perform the pathway enrichment to find pathways that were, affected by drug treatment and gain a better understanding of the underlying mechanism. And that's one workflow that we try to demonstrate how we reproduce and go deeper into the finding from Tahoe-100 million. And now we move on to the summary of our talk today, where basically in this session, we want to introduce you what is Tahoe-100. And then we want to show you how the data set look like on Talk2 data and how you can re-investigate the findings on Talk2 data. So in here, this is the summary. The Tahoe-100 is a very big data set with huge number of drug perturbations. And the Mosaic platform, the 50 cells in one wow experimental design, which was used for data generation, is great to minimize the batch effects. And then within Tahoe-100 on Tahoe data, we found out, or like we validate that the paraphenase effects varies based on different cell lines genetics or different mutations. And we also got the statistical analysis within the DGE to confirm the drug effects on marker genes. Okay, and that's all of our session today. In the upcoming weeks, we hope that we can, find more findings, find more insights from Tahoe-100 so that we can bring you more in the upcoming sessions. So, and that, yeah, I just want to head up to you about the next week or the week after session. We might still talk about Tahoe-100. So in order to, to have us to investigate this data set better, we would like you to answer some of the question in the, like, in the polls here. Let me just try to launch the poll. Okay. So now you should see the poll on the screen. We would like to ask you some further questions about, like, what type of downstream analysis you would want to perform on this data set, and what is your primary interest when using the data set. And last but not least, what kind of feature on top of the data would you find most useful for analyzing this data set? So please let me know. And also, I think this now is the Q&A time. Please let us know if you have any question. You can type down onto the Q&A box or even in the chat box, and we will answer that right away. Okay.
Yeah, we definitely appreciate everyone's feedback, especially since we introduced two kind of new concepts in this talk. We introduced the new data set, and we also introduced the new UI that will be released for Talk to Data beta as well. So we're open
Mhm,
to hearing feedback on that.
mhm. Okay. Thank you so much. Oh, I can see that many of you guys really want to have more the password enrichment. So yes, a good news that we are working on the password enrichment, and we hope that we can bring you some insights using the password enrichment in the upcoming weeks. And also we can see that, oh, this is nice because you want to use the Tahoe-100 for drug target identification. So yeah, okay. And and yes, this is one thing that we are actually trying to do at the moment. That's to integrate Tahoe-100 with our single cell RNA-seq database. So as you may know, we have a very huge single cell database with up to 200, 300 million cells. So we really want to incorporate that with Tahoe so we can revalidate the findings from Tahoe in our single cell RNA-seq database. So, so thank you. That's one thing that we are working on. Let me see here. Oh, we have one question from Pathak. Let me just this is off topic. Would Spatial X be the correct module on bioturing for spatial transcriptomics? And are there tutorial videos on this analysis? Oh, yes, yes. So Spatial X is now our main module for spatial transcriptomics analysis. And we actually have the guideline and also even the blog post to show you how to use Spatial X. Our college will put that in we will type down the link for you, and yeah, you can access that right away. Okay. We can. So I will open the floor for questions for around maybe 1 or 2 more minutes. Okay. Okay, so I think that's everything for today. thank you so much for joining us today, and we expecting to meet you in the upcoming weeks when we explore this data set further. Again, thank you a lot for being here with us, and see you later.
Thank you, everyone.
Yes, thank you. Bye.